Okay, so what we'll do is I'll talk to you about cotton. Remember though, in many instances you could replace the word cotton with quercus or any other sort of plant. So the approach that I take is basically, it's not particularly plant specific, but rather process specific. So I try and bring processes together rather than plants. So I'll tell you a bit about climate, present and future. It's probably a review for most of you. We'll look at the main and interactive effects of climate drivers, CO2 and temperature, extreme climate events, drought and floods, and importantly look at plant soil feedbacks, and then talk a bit about the future work. So these data are from Australia, but they could really be anywhere in the world. And so CO2 has been rising rapidly. I think it's now 407 parts per million. This is why. So if we look over the industrial period, we see that fossil fuel and industry have produced a lot of CO2 emissions, land use change, removal of land from forested areas and putting concrete on it. And then some of that CO2 can be taken up by the land sink. And some of that can be taken up by the ocean, but the difference between what's produced and what's taken up is what goes into the atmosphere, and that's why it's rising rapidly. Now we always talk about CO2, but that's just one of the greenhouse gases. So CO2 concentrations have indeed gone up, but so has nitrous oxide, which is a byproduct of a lot of agricultural operations, as well as methane and some synthetic greenhouse gases. So summed, their forcing power is over 490 parts per million now. So we can't simply focus on CO2. Now this graph always concerns me because if you look at the time period, so that line is basically the 50-year running average or 70-year running average. If you look at that line, temperatures used to be below that average, but I don't think it's been since about 1978 that the temperatures have been below that running average. And in fact, as you can see, they've become appreciably much higher than that running average. It also doesn't seem to matter as much whether the global circulation patterns in the ocean, such as the El Niños or the La Niñas. Volcanoes typically cool the Earth, but even that is not dropping temperatures below this running average. So we're certainly in a serious warming trend. And the climate has warmed consistently, both on land and in the oceans, which is important. And if you look at Australia, that big circle is where cotton is grown in Australia. But the mean temperature has gone up about 1 degree C over the last 100 years, and there's really no place where the temperatures remain the same or declined. And there are an increasing number of days with temperatures above 35 degrees C. That tends to be 35 C, I believe, was chosen for human health reasons, but we also apply it to agriculture and native plants. Pollen tubes don't form properly above 35 degrees and so on. So there are a lot of biological characteristics that are affected by temperatures above 35. And so what we do see is that we're seeing a lot more high temperature extremes are more common and cool temperatures are less common. So if we look at the daytime temperatures, we can see that 2 standard deviations, only about 2% of the time were daytime temperatures considered well above average, and now that's risen to about 11.5%. If we look over here at nighttime, yep. So here we had 2% and now we're up to 11%. If we look at the nighttime temperatures, 2% of the time they were much warmer than average, and now they're up to 9%. So not only are we getting shifts in the averages, but we're getting shifts in the extremes. And we're having far fewer cooler nights or daytimes and far fewer cooler nighttimes. So everything is shifting. And that means extreme heat wave events have become a lot more common. I'm sure your grandparents tell you at times, well, it used to be hot in the summertime. It was sometimes, but it's a lot hotter now. and it's a lot hotter more frequently now than it used to be. And so all of the metrics are pointing towards a more extreme climate. And one of the reasons that this is important is that if we look at something like vapor pressure deficit here, or the ability of the air to hold moisture, and we look at air temperature, and this is relative humidity, what we're finding is that for every 1 degree rise in temperature, the air can hold 7% more water. And that has consequences. The consequences are that it generates greater atmospheric water deficit in dry periods because the air can hold more water before it rains. It also means that it can generate larger rainfall events in wet periods because the air is holding now a lot more water. So what we're getting are longer periods of dry, and when that dry is broken, we're seeing really large rainfall events. And part of it is due to that. So it then seems to be, if you sum these things up, we have highly variable rainfall with extreme drought and extreme flooding. And those have been predicted by the climate models. And so if you say, oh, rainfall hasn't changed in Australia, true. It's unbelievably wet in the northern part, and it's unbelievably dry in the southern part. The difficulty for Australia is nobody lives here, but we all live where it's extremely dry, and all the crops are grown where it's extremely dry. And so the impact on the population is quite significant. So, to conclude this cheery introduction, global temperatures continue to rise. My apologies, gentlemen. Sea levels rise, temperatures are increasing, extreme rainfall events, more fire weather, which of course is very common in our area in Australia, and we're getting decreases in winter and spring rainfall, which are quite important. So we did a little bit of book work, and we looked at climate and extreme event impacts on key plant processes in cotton. We found 168 studies on cotton since 1965 from Glasshouses field that had one of these climate variables involved. So certainly elevated CO2 and warming have been looked at fairly heavily, as has drought, with a bit less on heat waves and flooding. And really VPD has only come into vogue more recently among scientists, although it's been important for a long time. But if you look at that, we see that, here are the processes. CO2 generally has had a positive impact. Warming can have a positive or negative impact depending on whether you're below the optimum or above the optimum. Heat waves generally have negative impacts, as does VPD, drought, and flooding. But we noticed that very few studies looked at multiple climate factors, and very few studies looked at climate extremes. So people tend to have their favorite process and their favorite treatment, and then they delve into great detail, but they don't look much beyond that. So our lab has taken a slightly different approach, and so what we've done is we've looked at these processes: growth and yield, the physiology of photosynthesis, stomatal conductance, transpiration. We've also delved now into plant hydraulics in cotton. Nobody was doing anything like that. We do a lot of work on the microbial function and community aspects, nitrogen cycling, and then something called legacy effects, which in the ecological literature is sort of known as ecological memory. So we've been looking at that as well. We also look at it across CO2, warming, drought, and then extreme climate events. And we have a few more X's, we've worked on that area. A couple more X's to add over the last year or so. And then Victor Rezko is a very influential fellow. So we've been looking a lot at the biological clock. That is, the control that the clock has on the processes of photosynthesis and conductance and so on. And pigment production, photochemical efficiency, respiration. Turns out the impact of the clock is much stronger than I certainly had thought. And hence we've been looking at nighttime water loss through transpiration and stomatal conductance. And in some areas in the deserts in North America, it constitutes about 20% of all the water loss is at night through supposedly closed stomates. Okay, so that means we've been looking at climate change Impact on crop productivity, as everybody knows, CO2 tends to increase plant growth, photosynthesis, and water use efficiency. Temperature will increase plant growth if you're below the optimum. It always accelerates the rate of development. And so what we did is we then took these factors and then we looked at cotton farming in Australia, which occurs on 3 different major soil types, which we selected 2 of them. But like all farming, it's a high resource input system. So there's a lot of nitrogen that's added to the system, and there's water or irrigation when you have water, which we don't have anymore. Water management is quite crucial. So the major threats to the industry are flooding and drought, and both occur somewhat regularly. Flooding tends to decrease root growth, nutrient uptake, and photosynthesis. And drought, of course, means nothing's growing, so there's that. So there's a lot of loss of crop productivity, and we're asking ourselves, what about extreme weather events? Well, this is one of our research facilities in Narrabri. That's my postdoc. She was my graduate student. CRDC is the Cotton Research Development Corporation. That's what a good cotton field looks like. Those facilities that you see up there have CO2 and temperature control, and we put them in the field, so we have field control of the environment. We can also alter the vapor pressure deficit in there, and then we can add water as we see fit or nutrients. So it gives us sort of more field realism. This, however, is images from the last 6 months where most of the reservoirs have dried up and cotton is not being grown, and a lot of the cattle are dying. There are towns that are literally running out of water. And towns, I mean 100,000 people, not 10,000 people. Cotton planting was down 45% this year. Yield was down 50%. So it's a very significant economic impact, which is being driven by climate. So what we did, I'll go through 3 case studies. We looked at elevated CO2, temperature, and soil water deficit on cotton, and we asked ourselves what the impact of warming and soil water deficit would have on the growth of the plant. Let's see if I can get this to work. So what we did is we had an experimental design in which we tried to replicate what happens outside. So we had a ambient temperature here, warm temperature there, elevated CO2 there, ambient CO2 there, elevated CO2 there. And then we would allow the plants to dry and then re-wet and dry and re-wet. So we'd go through 2 drying and re-wetting cycles so that plants would be more similar to what you'd see in nature. And then what we did is we looked at whole plant water use over the course of the season and mass production. And we found that under ambient CO2 conditions, what you'd get, or ambient temperature conditions, elevated CO2 increased the biomass with a small increase in water, which is what we've been told for a long time, that somehow elevated CO2 would be the panacea, that we don't have to worry because with higher CO2 under ambient temperature, that's probably true. But under elevated temperature, we didn't see a CO2 effect. We did see bigger plants, but we also saw significantly more water use. So under warmer, higher CO2 conditions, we will get bigger plants, but we'll also use a lot more water, which we don't have very much of now. So just to summarize the main and interactive effects of this, we found that elevated CO2 gave us bigger plants with higher photosynthetic rates, but also greater water use. Elevated temperature gave us bigger plants, but with much greater water use. When plants had water deficit, there was a certain loss of productivity. When CO2 and temperature were combined, they were the biggest plants, but we had reductions in water use efficiency. And then the water deficit took away the elevated CO2 positive impacts. And when you put temperature and water deficit together, there was very significant increases in plant water use. So overall, we sort of came to the conclusion based on this study that warming and drought will generally reduce the positive effects of elevated CO2. So the next thing we did is we wanted to look at the interactive effects of CO2 and temperature, but then we combined it with floods and droughts. And we also looked at the soil component. So we're going back to the same idea here. And so this was a fairly complex experimental design which took advantage of our research facilities. So in this instance, what we had was a, no worries. Here's our glasshouse. So what we did is we collected soils from the field, and then we drove them 5 hours south to Sydney. We collected the A and B horizons, and then we reconstituted the soil and let it sit. So we were using, so the right soil microbial community was in there, and the structure was as good as we could do. We used 2 soil types. We had 3 water treatments. We had a control, a flood, and a drought. On top of that, we added temperature, so we had an elevated temperature treatment, ambient and elevated. We then added CO2, so we had ambient and elevated CO2, and then we crossed them. So we had 2 soil types, 3 water treatments, 2 temperature treatments, and 2 CO2 treatments. And then we measured soil fertility and nutrient availability. We measured soil function, the microbial community, and then the physiology and productivity. And what we found was that under current conditions, we found that seed cotton production was increased, flooding had no effect under current CO2 and temperature conditions, but drought led to a 40% reduction in productivity. When we then added the additional future climate conditions, we found that flooding caused a 30% reduction under elevated temperature, and drought a 70% reduction under elevated temperature and CO2. So they both had very negative impacts. And that impact was greater on one soil type relative to the other soil type. In terms of the flooding impact, so those were the soil nutrients, what we found is under current conditions, we got a 75% reduction in nitrate, and under future conditions, we got a 90% reduction in nitrate. And nitrate's incredibly important in terms of cropping rotations and things like that. If we looked at the drought impact, what we found was that 30% of the nitrogen was unused, and under future conditions, 50% remained unused. So we did get differences in nitrate availability dependent on the treatment, and we'll see why this is important. So if we look at then a summary, so flooding tended to reduce productivity. It also, well, it had a small decrease in plant growth and soil nitrates. If we then added the future conditions, the impacts were much more negative. So under elevated CO2 and elevated temperature, the impacts were more damaging. And under drought, of course, everything was negative. And under future climate conditions, things were equally bad or worse. So in future climate conditions, what's the impact of these conditions? Well, both flooding and drought are going to generate a loss of net productivity, which I guess is not surprising, but hopefully we can tell you why that's going to be the case. So one thing that we then did is we looked at something called the legacy effect, and we looked at plant soil feedbacks. In order to give you some sort of sense, we have temperature and CO2, which have direct effects on plant growth. We have flooding stress and drought stress. So those can affect plant growth, but you also have flooding and drought impacts on soil moisture, soil nitrogen, and various soil processes. And of course, they affect these processes of photosynthesis and conductance and their linkages between what's happening below ground and what's happening above ground. And that's on any given day. Then the question becomes, what are the long-term effects that might be occurring over months or years? And that was our interest. So as everybody knows, Rich Norby's work in Tennessee, in forests, in face forests, have shown that CO2 increases productivity, so you get this nice stimulation. And if we stopped our experiments then, we would say CO2 is massively beneficial. But if you continue the experiment, you find that that's not always the case. And in fact, you get reductions in productivity that occur after 3 or 4 years. This is in a natural forest that has been fumigated with elevated CO2. And Ichilo came up with some explanation, which he derived from a bunch of people, but in essence, the idea is that you get progressive nutrient limitation, in this case nitrogen, which says that yes, if you have your resources, you can take advantage of the elevated CO2, but Liebig's law of the minimum would suggest that if you have something else that's limiting, then your stimulation is going to be reduced. People have put this forward for other resources as well, water, phosphorus, and so on. So why does that happen then? Well, part of it happens because if we look at the cycle here, we can see that elevated CO2 increases net primary productivity. That increase in primary NPP generates an increase in carbon input to the soil, which changes the carbon-nitrogen ratio. Also, nitrogen is sequestered in the biomass and in the litter. In turn, that means more nitrogen is sequestered in the soil organic matter, which then, due to competition between the microbial system and the plant, you get reductions in nitrogen availability and reductions in nitrogen uptake and labile soil nitrogen. So this is sort of how that operates. Now the question is, so that's nature, but then what happens in a cropping system? Can we overcome this progressive nitrogen limitation or limitation in resources? So there's a large external nitrogen input, and the question is, can we sustain productivity? In order to understand that, we have to look at plant soil feedbacks and their long-term impact on crop productivity. So what we did is we took that first year's experiment and we took all the data from that, and then we generated what we would call the legacy effect. So just to recap, we had these plants, we fertilized the soil, this is what a farmer would do. We put the plant in a pot. When I say pot, I mean a really big pot, so it's not really limited. We then grew the plant up for a season in the different environmental conditions. Then we collected the cotton, we harvested the cotton, we harvested the seed, and then we cut the plant as you would in a cotton farm and put the residue back on the field as they do. And then we let it lay fallow in our glass house under winter conditions for 7 months. Because that's a simple experiment, we decided to make it complex. So what we ended up doing then is in the second season we added fertilizer again to the soil that already had a full year of treatment. We put a new cotton crop in, and then we grew it for the second season. And what we then did is we looked at the responses in the first season and compared the responses to the second season. And so this is a response ratio where everything on this side is a positive response and everything over here is a negative response. And what we found in the season is, in general, elevated temperature increased vegetative growth in seed cotton yield, and CO2 also increased growth in yield via increased photosynthetic rates. But if we then add the second season to that, we didn't get the same results. What we found is that the magnitude of the climate change impact was reduced. And it was reduced because what we got were reductions in seed cotton yield, and we got less of a stimulation in photosynthesis due to lower leaf nitrogen. And so the yield was reduced, leaf nitrogen was reduced, photosynthesis wasn't as stimulated, and the question is, why did that occur? And we asked ourselves, is it a natural nitrogen limitation that's happening? Well, what we ended up doing then is we ended up looking at leaf nitrogen concentration as a function of soil nitrate at pre-planting. And everything here is elevated CO2, and this is ambient CO2. So elevated CO2 reduced soil nitrogen, and that led to reductions in leaf nitrogen. And leaf nitrogen was driven by soil nitrogen. And then we saw that seed cotton yield was strongly related to leaf nitrogen. So the impact of changes in the soil nitrogen affected the leaf nitrogen, which in turn affected the yield. But oddly enough, seed cotton yield was not related to photosynthesis, which causes me to weep because photosynthesis is my favorite process. So I was thinking, as most people do, that increases in photosynthesis will drive increases in productivity. There are just far too many variables that play into that. So that was not the case. So what did we find? Well, overall, we had the first season, we had the second season, and not to bore you with all the details, but soil fertility recovered during the fallow in both the control and the flooded fields because we got mineralization and we got nitrification, but there was certainly less recovery under elevated CO2, particularly at ambient temperature. And you must note that we added the same amount of fertilizer in the second season as we did the first season, so the impact of the actual nitrogen in the soil played a very large role in determining what the yield was. So the question is then, since we were able to demonstrate that seed cotton yield is a function of leaf nitrogen, that there was a very strong relationship. And so we tracked, as I had mentioned before, we tracked soil nitrogen all the way through all of the different seasons, and we found that it was a function of, leaf nitrogen was a function of this starting nitrogen, which in fact in the end ended up driving yield. So there was some sort of legacy, there was a legacy effect that occurred. So we're trying to figure out what was driving that legacy effect. Why was the soil nitrate availability different in the second season compared to the first season? And so we looked at the soil microbial community, primarily the functional aspect of the soil microbial community, to try and determine whether that was driving soil nitrogen availability. And so if you look at soil nitrogen, or nitrogen cycling in the soil, it's made up of a bunch of different processes, including what you have, the litter input, which is affected by the CO2 and the temperature, root exudation, of course, the sugars that are exuded, and then volatilization or leaching of the important nitrogen components, and then denitrification, the gas loss, and nitrifier denitrification. Now, all of that has to, so in order to understand that, you have to get a better idea of what the denitrifying community is doing and the various oxidizer communities are doing, and we have that capacity to do that. And then you have to look at the soil microbial community as well. Oops. And what we found, in fact, was that the litter input was increased and root exudation had been increased due to these different elevated CO2 and temperature, which in fact ended up increasing the soil microbial community, which increased the competition then that these microbes had with the plants, which generated a reduction in plant available nitrogen, which meant that nitrogen was not as available to the plant as before, which means even if you add nitrogen to your system, that the role that the soil microbial community plays in nitrogen cycling can overcome to some degree that extra nitrogen that you've added into your system. And of course this applies to all ecological systems. So what did we find? So we found that there was a positive response to elevated CO2, but that was negated by warmer temperatures. Temperatures increase plant water use, and that's incredibly important. When I say plant water use, I mean whole plant water use, not leaf level. People get confused about that. You can have lower leaf level water use, but if you have more leaves in a bigger plant, you still use more water. Flooding and drought decreased productivity. We had some interactions between CO2 and soil nitrogen limitation. There are obvious links between the nitrogen and the cotton yield. No relationship here. But I think most importantly, we found these legacy effects in plant soil feedbacks were the key regulators of the plant response. And so I gave a talk at a Gordon conference recently where a bunch of people have gotten together. The Gates Foundation funds a lot of this work, and the idea behind it is to increase food productivity. So there seems to be an immense amount of work, and I don't know what's going on in this realm here, but an immense amount of work about increasing photosynthesis as being the key to increasing food productivity, which includes things like inserting C4 genes into C3 plants and so on. But I guess my argument when I was there, because I was slightly on the other side, and I basically said, a lot of this work's being done under optimal conditions, but we need to be able to consider the main and interactive effects of CO2 and temperature. If you're inserting C4 genes into C3 plants, that's not particularly valuable if the CO2 concentration is now 500 parts per million. What have we gained? C4 is a concentrating mechanism. So each year the CO2 concentration goes up, the value of turning a C3 plant into a C4 plant declines. In 50 years, will that still be valuable? That's a good question to ask. Also, all the benefits that you may or may not accrue from warmer temperatures and higher CO2 may not be there if you have increasing droughts and floods and heat waves. That'll always detract from the obvious goal of increasing carbon production. We really do need to prioritize reductions in whole crop water use and nutrient fertilization, and we need to account for plant soil feedbacks on the microbial community and on nutrient cycling. A lot of us work in silos, so we're plant people or soil people, and obviously there are groups that get together and have conversations and work together, but that's really a strong focal point in our lab group to be able to do that. And so we need to be able to generate a better mechanistic understanding of these relationships if we're going to increase yield. So this was a lot of people did a lot of work from a lot of different universities. And so I'll take questions on this, but I'd like to just, if I have a few more minutes, can I talk a bit about the hydroponic work that we're doing? Okay. So we are now a cooperative research center for something called the Future Food Systems. The government of Australia has a large investment program that they put into things that they consider important. So often it's robotics and engineering and things like that. But the government recently gave us $40 million, and then private industry kicked $140 million in. So there's about $180 million investment over 10 years to try and increase food production. Now that includes things like the utilization of robotics, environmental control systems, which is our specialty, improved breeding programs, generating greater nutritional value over what we produce, pest management, which moves away from organophosphates and goes more towards biologicals. And then the idea is to grow the plants close to the consumer. So Australia is a very simple place to do that. There are 6 cities in the whole country. So if you grow food just outside one of the capital cities, you can get it to market in 2 hours. So that's the idea behind it. So we've taken a protected cropping approach. This is our facility. But basically the idea behind it is to maintain food security in response to climate change. Like I said, cotton production's down 45% this year. Grains have been down quite significantly. And so these weather impacts are quite substantial on food. Reduce our carbon footprint by having lower transportation costs. We have input solar power in. So what we do is we offset the cost of running our facility through solar, and we're increasing our photovoltaic, semi-transparent photovoltaic uptake. So when the light comes through, it actually is converted to energy. It then comes through, powers photosynthesis. We use less water, we use less nutrients, and so on. We use biological control systems, so we allow the beneficial bugs to take care of as many of our pests as possible. So we're proactive about that. We use natural pollinators. We have stingless bees that move through our place. We've got hives set up in there, and so the bees are able to do pretty well in terms of pollination. If you do cropping work, pollination is key and very expensive. And this novel glass technology is which is what we work on. So we are part of the national, we are the National Vegetable Protected Cropping Center. This is our glasshouse. No, it's not that large, but it's here, so it's pretty big. These are some of our other glasshouse facilities, and we're expanding our footprint for horticulture. We're producing a facility that's maybe 10 times this size now on campus. So we're getting into it in a very large way. The idea is to have demonstration bays, training bays, research bays. We have full control of temperature, CO2, humidity, and so on. And the idea is to maximize productivity and minimize costs. So the glasshouse operation is pretty high tech. The point behind it is that it's a research facility, so put the best stuff you can into it. We know in 10 years it'll be obsolete, but at least we have very good control of CO2 and temperature and nutrient delivery systems. Each plant has an actual nozzle that goes to its base, so we can direct water and nutrients to it. And then, of course, we have these various control systems. But the idea is, because the production of plant material in protected cropping can be quite expensive, particularly in a place that gets a lot of sun, like Spain or like Australia, we need to be able to reduce this radiation load to decrease energy use. So the idea is to increase energy use efficiency without losing productivity. We use something called a smart glass. It turns out to not be as smart as we'd hoped, but what it ends up doing is reducing shortwave radiation and IR. And the idea behind it is it's supposed to block the light that contributes heat, but not photosynthesis. So we coated 2 really massive bays with this smart glass and then 2 bays without. The manufacturers told us this is what was going to happen. Less light, photosynthetic light would come in, but it would be good otherwise. And that it would block IR, that high heat producing light, and UV. And that we were not to have any effect in the wintertime, but we'd have a big effect in the summertime, positive effect. So we set the whole place up with the big sensor systems so that we could actually get a much better idea of what's happening inside the facility. So we have light, air, and nutrients. These sensors are arrayed spatially throughout each of the different bays from top to bottom and across the sides and integrated. And we measure the short wave and the long wave radiation. So the end result of having all of these sensors is that we do have a lot better idea of why our plant productivity has been altered. And what's happened is that in the UV, so this is the control, that's green, at the roof and the wall. Let's just worry about the roof. So the idea is that the control, that 70% diffuse light, it's the best glass you can get from the Netherlands. It's very good, very productive. We then coated that glass. So what happens is that will let in UV, but our smart glass doesn't let in UV. And also we cut out this IR out here. Unfortunately, we also cut out some of the photosynthetically active radiation, which is a problem. And it's a problem because the different wavelengths of light have various roles, but UV is important in pigmentation and also protection against insects. We also found it affected the dermal production, leaf surface production. And then when you get into the red, you've got a lot of flowering, which is really important. So what we did is over time, we basically were trying to optimize this glass. We were able to, this is the control and this is a smart glass. We reduced the amount of shortwave radiation by 53%, but we also reduced PAR by about 20%. So we were saving some energy, but we were also having a negative impact on photosynthesis. So we did a couple of trials, took a lot of different measurements, and in the end what we found is here's a light response curve. That's photosynthesis, that's light, and then if you look at the control plant, the average light coming in is 1400 micromoles, and this is the output. If you look at the smart glass, it's down to about 11, and that's the output. So not only reducing the amount of light, but we're reducing the efficiency with which the light is being used. So it's a double whammy. So we've actually changed some of the characteristics of the leaves and the photosystems via this, and that in turn has generated reductions in carbon assimilation. The disadvantage, of course, is that with less carbon, source-sink relations say that you can produce a bunch of sinks, but with crops, what happens is you can produce it, but if there's not enough carbon to drive it to maturation, the plant drops it. So we end up doing fine until we get to the fruit part, and then the carbon loss that we get by the smart glass means that those fruits are aborted and they don't go to full production. It's just classic physiology, ecology, nothing novel there. But the fact is that what we end up getting is a 32% reduction, which we repeated the experiment and it was 24%. So, and that had largely to do to a source-sink imbalance. We've since employed a couple of postdocs whose specialties are carotenoid biosynthesis and also stomatal physiology and membrane physiology. And they've showed us that our stomates slow down under smart glass. They're not as rapid to respond to changes in the environment, which means that the plants don't take advantage of the good opportunities. We've had some fairly significant changes in the carotenoid biosynthesis as well, which has reduced the efficacy with which these plants use light. We didn't affect the fruit quality pretty much. And so we went in and we looked at metabolites, and we only found changes in a fairly small number of metabolites that we followed. So only 16 out of the 408 that we tracked had differences. So it means that not a lot is changing in terms of the fruit quality. But what we ended up doing is we found that we got a 53% reduction in the shortwave radiation, which generated an 8% increase in cooling efficiency. And it also gave us an 18% increase in nutrient and water consumption. So the smart glass did have some advantages in terms of energy use and in terms of water and nutrient use, but it also reduced productivity. So we've rerun these experiments on capsicum and we're finding somewhat similar results. So, final slide, the smart glass blocked the light contributing to heat, but it reduced PAR, reduced photosynthesis and yield, and I think it's a source-sink relationship issue, which we're now delving into. Some basic changes in sort of the fundamental plant physiology. So we're going to re-engineer the smart glass, so we're working with engineers now to make sure that the characteristics allow UV in, which is valuable biologically, and we have to be able to increase red light penetration, and we can see if we can cut more of the IR out. And that's why we're working with the engineers on that. And we're trialing different crops as well. And that's just the nuts and bolts. Okay, thanks.
Okay, so two very different sort of halves to the talk, but I guess it's nice to see the two sides, because it's not just the science, it's also looking forward in terms of what do we use this knowledge for and how we optimize production in these systems, taking into account this world that we live in. You know?
Yeah, and I think another thing that's lost occasionally is people become wedded to particular ecosystems or to particular crops, and they work within that. And I think one of the things that we've discovered is that you can move across ecosystems and you can move across crops, but photosynthesis does not change. Stomatal conductance, transpiration, the physics and chemistry, microbial communities, these are all relative constants. And so you should be able to learn from one system and apply it to another.
Okay, questions from the public? Yes?
With the smart glass not giving the result that you were expecting, do you still believe it's a valuable system?
Yeah, yeah.
Why?
It is the future, I think, of crop production under plastic or glass, because Spain is probably the largest user of plastic in the world and one of the largest protected cropping places in the world. And so we have to make sure that what we do uses the least amount of energy and increases water and nutrient use the best. the reason we did this study is that the industry was ready to just slap this up on all of their glasshouses. But I think what we're saying is that we really do understand where it is efficient and where it's not efficient. So would I put this in my glasshouse? No. Would I put a variant of this in my glasshouse? Absolutely. Yeah. This is the same stuff you get on buildings around the world. You know, that light blue tint that you see, most office buildings have some form of this in them. Now, in an office building, we're not worried about the photosynthetic rate of the plants, and human beings are just trying to save energy, whether you have UV or not. We don't want UV, and we also don't want the heat. So it's perfect for an office building. But for a growing facility, it needs to be modified.
Jordi?
Thanks for the nice talk. So I have a question on the cotton side. You showed that there is a connection between soil nitrogen and leaf nitrogen, and then between leaf nitrogen and yield. And then you show that this is not through photosynthesis. But then my question is, what is the plant process that is affected? I mean, that it's driving this effect of leaf nitrogen and yield, which is uncoupled to photosynthesis.
Yeah, so the driving force, I think, is, well, one thing to note is photosynthesis is very high in cotton. So it may be that the photosynthetic rates are already relatively high. And so carbon assimilation is not really a limiting factor in terms of production in these systems. It's how you allocate your carbon.
But then this would be affected by leaf nitrogen. So allocation would be a function of leaf nitrogen somehow. I mean, according to your result.
Yeah, yeah. Well, we're still perplexed a bit about how there's essentially no relationship between carbon assimilation and the yield. But I think a lot of the productivity, so cotton is a bit tricky. So only about 2% of the productivity is in the actual fiber itself. And what happens in the fiber is only important for over about a 2 or 3 week period. So one thing I think may be happening is that when we take our photosynthesis measurements, we have to be a lot more specific about when we take them and how they relate to the development of the fiber. Yeah. But I share your perplexion because, but I was willing to put it up there anyway.
Yeah, also in this line, how do you reconcile the fact that microorganisms are carbon in the soil, are mostly carbon limited, with your explanation of the role of nitrogen there? Because it seems in mostly your conclusion is that it's mostly because there is a lack of competition for nitrogen, but microbiota in the soil is mostly limited by carbon. So all this first part, the connection between carbon, CO2, nitrogen, there is something that also that leaves me a little bit perplexed, as you say now.
Right,
yeah?
Right. So microbial ecology is not my specialty. But my understanding is that when you alter the carbon-nitrogen ratio and you then decrease the amount of nitrogen that's available, that you've got competition for that nitrogen gets immobilized. Is that not correct? The nitrogen gets immobilized. So when you've altered that ratio, you immobilize the nitrogen, which makes it less available to the plant. Correct? Okay. So reiterate your point then, to help me out.
Yeah, but then what, in fact, the drive microbiota is the carbon limitation in the soil. This is something that the whole community is not well aware of.
Yep.
Plants are really nitrogen limited, but microbiota they are not usually.
Or not
They
nitrogen.
need the substrate. You only need to look at the stoichiometry,
Right.
carbon nitrogen of plants, liter and microbiota. Then you
Right.
realize that they have plenty of nitrogen. Microbiota.
Okay.
So there is something there, but in fact our results and our way of looking, let's put this is a particular thing.
Okay.
But globally, I think that we fully agree with you, and our global results,
Right.
they make us think like you,
Right.
that even if we have a fertilized wall with CO2,
Yes.
then we don't have water enough, or we don't have nutrient enough.
Right.
So this fits very well the let's say, global ecology.
Yeah.
Yeah, but the mechanism is what drives us crazy.
Yeah.
Find out what's going on there.
Yeah, I'm with you on that one.
But at least your experiments fit very well with, let's say, what in global ecology is done.
Yep.
People is looking at what happened in the last decades with increasing CO2,
Yep.
and at the same time we follow what's going on with productivity,
Yep.
and then what's going on with what we call the CO2 fertilization efficiency.
Right.
Yeah? It's decreasing, as in your experiments, very much what we are seeing globally.
Yep.
And so it would be very good that you would help us to understand the mechanisms behind this role of water. For example, because with elevated CO2, we always claim that there is an increase in water use efficiency, and then some people think that this is going to overcome the problem of drought, but we continually see that is not the case.
No. Well, and I agree with you on that, and I think a part of it's because we have taken the photosynthetics to models, and those will tell you that you will get less water use per amount of carbon fixed, but that doesn't tell you what the whole plant is doing. Right. And so I see it often, even in papers I review, where the first sentence is always, you know, we're going to save water under elevated CO2. But that makes assumptions that the leaf stomatal model applies to the whole plant. And what we find is that leaf area production is increased dramatically. And so if you do save on a leaf area basis 10%, but you have 20% more leaf area, then you've got more water use.
Yeah. We have to talk about that. And then also about your experiments, how do you measure the root exo rates? Because we know that Catherine is not here. No? But, I wonder, and we wonder, how do you do? Because we always discuss which is the best method. This is a methodological question.
Okay.
Yeah? How do you measure carbon exudates?
It's very hard to do. So Braj Singh's lab at our university is largely responsible for measuring the exudates. So I'm not really that familiar with the exact techniques that he uses, but it would be in our papers. But yeah, we do. So for instance, though, when we do our watering, we collect all of the extra water and all the material that comes out of the pot, and then we analyze that too. So in essence, we create a closed system by doing it that way.
Yeah, because we always discuss which is the best method, because most of them, they are quite disturbing.
Yep.
It's very, very hard to find out what we are really doing.
Yeah.
Without disturbing the system.
Yeah.
So we do with solid substrate, we do with water substrate, water solutions.
Yeah.
We do at the end also.
Yeah.
We are never sure what we have.
Well, we were very careful in this particular study to use the native soils. And we were so we went into cotton fields and we collected their soils and we recreated those horizons as well. I think an awful lot of studies that are done in controlled environments use sand, they use peat, they use dirt, and they don't really take into consideration the existing microbial community and the soil chemistry and the structure of the soils when they do their experiments. When we did our review of the literature, we had a really hard time finding people who even told us what type of soils they were using. So I think there's a real miss between the physiological community who think that as long as the plant's been grown, it doesn't matter what it's grown in, and the soil community who obviously take great pains to worry about that. Yeah.
So thank you very much, because we learn a lot.
Well, I'm still perplexed about some of the connections myself.
We are perplexed.
Yeah, exactly.
And we also, here in CREAF, we also are going a little bit in the same way that you are taking in your research, because after nature, we also care about humans as part of nature.
Right.
And so we are wondering about how do we apply all that we are doing also to food security and all these things. So keep in touch.
Yeah. Yep.
Any more questions? No?
Okay.
But then we thank you for
Okay.
very much for your
Yep.
talk again.
Good. Thanks a lot.